Hey, everybody. So the purpose of this presentation is to cover proteins and nucleic acids, which are the last two of the four biomolecules that we discuss in Unit 2. Um, kind of tacking on to the carbohydrates and the lipids that we've already discussed previously. So proteins are really the most diverse group of biomolecules. You can argue that they're also the most important, but it's kind of an interesting argument to have because you really need all four. All four serve a pretty specific function and can't really operate without the others. Proteins, for example, can only be made with the help of DNA and RNA, so they're really dependent on nucleic acids. But proteins are definitely the most diverse of the groups of biomolecules that we discuss this unit. So they're involved in nearly every dynamic function in your body. And like I said, super, super diverse. They have various functions such as enzymes, which we're going to spend a pretty significant amount of time discussing. Enzymes will have their own presentation and then also a kind of accompanying lab that goes along with it. They can also function to transport substances across the cell membrane. So transport proteins can either be embedded and pass right through the cell membrane, or they can be on the periphery, either outside or inside the cell membrane. We'll talk a lot more about that in Unit 4. They also function in defense, or like antibodies that function in the immune system to help fight off infection. They can serve as hormones like insulin, which is a signal protein. Again, we'll talk more about that in Unit 4. Receptor proteins, which can, to use the term, they can receive chemical signals. They are typically embedded in the cell membrane and have a binding site where different substances can bond to them that'll trigger some sort of chemical reaction inside the cell. Also movement, right? So contractile proteins, which aid us with muscle movement, things like actin and myosin in our skeletal muscle. And then structural proteins like collagen or keratin that help to provide structure and support in places in tissues like our skin. And then you have storage proteins like albumin in egg whites, right? So proteins can also serve as an energy source. It's not ideal. It's not the go-to energy source for a lot of our cells. Typically, that's going to be carbohydrates and then lipids. Proteins are almost like a last resort, but they can be, really, really beneficial, in terms of longer-term energy sources. So just to organize those functions and get them kind of all into one spot: structure, like collagen; movement, like actin and myosin, which we mentioned earlier; defense, which are antibodies that help to fight off infection; storage, like the albumin that we discussed, you can find in egg whites. And by storage, we're really referring to like energy storage. Signaling, so they can function as hormones like insulin or growth hormone, and then they can function as enzymes where they'll catalyze reactions. And we're going to talk about a whole host of enzymes. So it's interesting that the hair, right? The hair, horns, and then silk. So these are kind of the next three slides. Are all really made of the same protein. They're made of a protein called keratin. It's the same stuff that's in your fingernails. It really just, the reason for the differences, right? So you think of like that bighorn sheep's horns and your fingernails, okay, there's some similarity there, right? They're both rigid. But then you look at the hair on that dog and you'd think, it's made of something totally different. And then the silk in that spider web, no, there's no way that these are related. It really just has to do with how densely packed the keratin fibers can be and how they're interwoven. But the spider silk should show you the tensile strength, like really the strength of those protein fibers. And so that's just an example of a structural protein, right? Keratin. All right, so they're composed, now getting into kind of what they're made up of, composed of differing arrangements of a common set of 20 amino acid monomers, right? So monomers, remember, are the individual subunits that make up proteins, right? Polypeptide chains. Just like carbohydrates are made up of monosaccharides, lipids are made up of fatty acid tails, proteins are made up of amino acids. And there are 20 different amino acids that can be interlinked, just like there are 26 different letters and depending on how they're linked to one another and the sequence of the chain that they make up, right, they would have a different meaning. Same thing goes for proteins. So the functions of different types of proteins are really going to depend on their individual shapes. Here's another kind of throwback to that really important essential question for this unit, which is how does structure relate to function in living systems? Proteins are the prototypical example, really awesome example of this, because depending on the sequence of amino acids, they'll be shaped differently, like their three-dimensional structure will be different, and depending on their three-dimensional structure, they'll fit differently, and so they'll have different functions or different things that they can technically interact with. In this super important process that we're going to investigate during our enzyme lab in this unit called denaturation, it's just a fancy way of referring to a protein that has unraveled and lost its shape. And when it loses its shape, like we just said, that has a really big bearing on its ability to carry out its function. Right? So super important question to think about in preparation for any assessment in this unit. Why would a denatured protein no longer function normally? Right? Not a bad idea to kind of pause there and start to think about how you might piece together an open-ended response to a question like that. All right, so that's a three-dimensional model of an enzyme. Kind of doesn't really have a whole heck of a lot of meaning to us like that. But here is, a kind of more filled in, more detailed structure of that same enzyme. And what you're seeing here should be more familiar to you because we've practiced with the molecular modeling kits. Every time you see one of those blue spheres, that's a nitrogen. The black spheres that you see all over the place should be familiar because those are carbons. The white ones are hydrogens, and then the red ones are oxygens. So this really lets you kind of make that connection between atomic and molecular. And then there's some purple ones in there that are going to symbolize sulfur. Because, I apologize, the yellow ones in here are going to symbolize sulfur. Sorry about that. So that should kind of help you to make that connection between atom or the atomic level and then how they're bonded together to make molecules. And this is really what we're talking about when we talk about three-dimensional shapes. And it turns out with this enzyme that in this space right here, this groove, that's actually going to be known as the active site. That's the place where the molecule that the enzyme bonds to, that's where it's actually taking place. That's where the connection is happening. So the molecule that's going to bond to this to this enzyme is called a substrate. And once it actually bonds to the enzyme, let's just kind of finish this off here. Let's say that's the substrate. This is now referred to as an enzyme substrate complex, which we'll break this down a bit more in the enzyme presentation. But it's just to give you an idea as to the three-dimensional structure of a protein and what it could look like. So proteins are made from amino acids that are linked together, right, during a dehydration synthesis reaction to form a peptide bond. This is actually the example that's used in that dehydration reaction that we watched earlier in the unit. And like we said before, the function of the protein is really dependent on the sequence of amino acids that are placed in a chain when forming this protein. That's going to determine how they're actually folded together. So an amino acid contains really one unique bit and then three that are the same across the board. And the ones that are the same across the board are the amino group, right, which is nitrogen containing, the carboxyl group, which is that COOH group, and then a hydrogen atom. Those are the three bonds which all come off of one central carbon. So if you have that carbon here, right, typically the hydrogen is depicted as going up in that direction, and then you've got two additional bonds, one of them to an amino group, right, and then the other to that carboxyl group or the carboxylic acid, right? All amino acids have that same basic structure, that central carbon bonded to those three things. The only place that they differ, the one thing that sets the 20 amino acids apart, is that fourth bond that we say is to an R group, which is just kind of like a placeholder for variable. And depending what that fourth bond is to, that's going to determine which amino acid you're dealing with, right? So like we just said, the R groups are going to distinguish those 20 amino acids. Each having a different property, and that property is dependent entirely on the R group. So if you have a chain with a hydroxyl group, it's very likely that that R group is going to be polar. And so this amino acid would be considered a polar amino acid, and it's going to exhibit those polar properties, and likely it's going to want to interact with other polar amino acids or amino acids that have polar R groups. Right? So this is kind of what I mean. Leucine, for example, is hydrophobic, and this makes total sense if you recall back to the way that we discussed lipids and what makes them hydrophobic, right? Is just the fact that they're chains of hydrocarbons, and carbons and hydrogens typically get along really, really well. They share their electrons pretty evenly, and so there's no separation of charge. There is no positive or negative regions. But then you run into serine or aspartic acid, and these are going to be hydrophilic, and the reason being because of that oxygen, right? So we know that oxygen is more electronegative in relation to carbon, and that hydrogen kind of really gets easily bossed around by electronegative atoms. So that oxygen is going to unevenly share its electron with the hydrogen, and because of that, it's going to make itself or the region around it partially negative. And then all regions that are kind of losing out on those electrons are going to end up becoming partially positive. And just like water, right? Just like that happens in the relationship between the oxygen and the hydrogens in water. And so this R group on the serine molecule is going to end up interacting with water because its partially negative region will be attracted to the partially positive region in the water, and vice versa. Right? Same thing goes for aspartic acid. Some will actually have, these side chains that are, acidic. We're not going to really get super into that, but an acid is any substance that freely gives up hydrogen ions, or H+. And you see that that's kind of what happened here with the aspartic acid. That oxygen flat out stole an electron, giving up an ionized hydrogen, which is now just an H+, or really just a proton. And then cysteine is super interesting because cysteine has sulfur in its R group, and sulfur has these unique properties which allows it to form these different kinds of bonds that we call disulfide bridges, and that's going to inform what's called the tertiary structure of proteins, which we'll get to in just a bit. So it's just important to remember cysteine because of that unique characteristic. The fact that it has sulfur in it, in its R group, is going to influence the three-dimensional folding of that overall protein in very specific ways. All right, so amino acid monomers are linked together during that dehydration synthesis reaction, where you're basically joining the carboxyl group of one amino acid to the amino group of the next amino acid. So, if I just take a couple of seconds here and draw this reaction out, I know that there's figures that'll do it better, but just to actually see it happening, right? So the R group, and then here's amino acid number 2 with its 2 hydrogens coming off of its amino group, carboxyl group on this side, and then the R group. And we're just disregarding the R groups right now because we're trying to talk about linkage. So what's going to happen is that this hydroxyl group here, right, on amino acid number 1, when brought close enough by the actions of an enzyme, this oxygen is going to interact with this hydrogen in a way that's favorable to the oxygen, right? It's going to end up forming a covalent bond with that hydrogen atom. Again, it works out better for the oxygen than it does for the hydrogen. But what's super important about that is, yes, you form a water molecule, but more importantly, you've now got an unshared electron that carbon has and an unshared electron on that nitrogen. The enzyme bringing them together is now going to make it possible for the carbon and the nitrogen to covalently bond with each other. And this right here, that's the peptide bond, right, that we're talking about. It's just a specific name given to this particular bond that happens between 2 amino acids. Right? Additional amino acids could then be added to, like, that amino acid number 2 that we had in the drawing to create a chain of amino acids that we would call a polypeptide, poly meaning many, peptide referring to the bond that links them, right? So then, just kind of using what we just talked about, what process then would you need to undergo in order to digest the proteins that you eat into their individual amino acids, right? So food labels will say, the dry weight of protein that's found in that particular food item, but it's not like you're consuming the proteins and they're ready made and you're now going to use them. You're consuming the proteins, but then you'll end up digesting them into their amino acid monomers and using those amino acids to make your own proteins, right, that your cells need in order to function. Okay, so this is that figure that I was talking about. Does it much nicer than I do, but it was just really so that you could see it in action. This guy right here is just known as a dipeptide, di for two, because it's two amino acids that are linked by one peptide bond. As soon as you were to throw that third amino acid, then you would call it a polypeptide. All right. So a protein can have four, and this is where proteins can kind of get a little bit tricky to understand, but this is really kind of hammering home that notion of structure and function. So a protein can have four levels of structure. We refer to those levels as primary structure, secondary structure, tertiary structure, and then quaternary structure. So protein's primary structure is really just the sequence of amino acids in its polypeptide chain. So if I have, a glycine, and that glycine is bonded to, an aspartic acid, which is then bonded to a valine, and I'm just shorthand drawing these as circles, right? This just basic sequence, that is referred to as the protein's primary structure. Now, what's going to happen is that these amino acids are going to kind of interact with each other. Like we said, nonpolar amino acids are going to interact with other nonpolar amino acids. Polar amino acids will interact with other polar amino acids. The nonpolar R chains are going to influence the twisting of the chain because they're not going to want to interact with like the aqueous environment in a cell. So just the sequence is referred to as the primary structure. But then once those amino acids start to interact with each other in their environment, that's going to create the protein's secondary structure, which is really the coiling or the early folding of that amino acid chain. And this is really driven by hydrogen bonding. Hydrogen bonding is a really, really simple, weak kind of bond that takes place between hydrogen atoms and then atoms like oxygen or nitrogen that are in these amino acids. And then once that starts to happen, and you start to take on this early rudimentary three-dimensional shape, what can start to happen is that the R groups of each adjacent amino acid, or even of amino acids that aren't quite adjacent to each other, they'll start to interact with each other. And those interactions are usually a bit stronger than just the simple hydrogen bonding that starts to coil the amino acid. And we call that the protein's tertiary structure, the development of the protein's tertiary structure. And this is really the overall three-dimensional shape of a polypeptide that results from the interactions among the R groups. And like I said, these are just a bit more, I don't want to say permanent, because we talked earlier about denaturation and the fact that proteins can lose their shape. But in an ideal world where the protein is able to function, these are the things that are really keeping that protein held in place. And so a functional protein has to have this tertiary structure. Some proteins, right, not all proteins, end up displaying what we call quaternary structure, and this really just depends on the complexity of the protein. These are any protein that are made of more than one polypeptide. So here you have multiple chains, right, of amino acids that can interact with each other. Right? There are proteins that can be made of 4 amino acid chains that are complex together, or 6 amino acid chains like hemoglobin that are interacting with each other to make this overall three-dimensional structure that can execute a certain function. Right? So not all proteins end up being quaternary proteins, but to be functional, they do need to exhibit secondary and ultimately tertiary structure. Okay, so if a genetic, this is super, super important because this is a way to practice explaining that connection between structure and function, right? And this is a really big, broad, kind of multi-concept question. If a genetic mutation changes, first of all, what is a genetic mutation, right? And we haven't gotten to nucleic acids yet. But if a genetic mutation changes the primary structure of a protein, how could that destroy the protein's function? So just by affecting the protein's primary structure, how are you now impacting that protein's ability to function? So you'd have to think about, how does primary structure affect secondary structure? How does secondary affect tertiary? And then how does that affect function? Really, really great practice question, right? So I like this figure because this figure is just a visual way to understand what we were just talking about with the different levels of protein structure. So that would be a protein's primary structure. This is just an example, right? Just a long sequence of amino acids. I also like that it gives you this kind of zoomed in, actual, realistic look, right? This right here is one amino acid, and it's peptide bonded to this second amino acid, right? That's what's actually happening here. But that gets really, really cumbersome to draw. And so we just simplify amino acids by drawing them as like little spheres. Right? So the primary structure is just the long sequence of amino acids. The secondary structure is what starts to happen when adjacent amino acids interact with each other and are linked tentatively by these weak bonds that happen between hydrogens and oxygens largely. Right? Nitrogen can also hydrogen bond, but really it's driven by the interactions between oxygens and hydrogens. So that's secondary structure, where you can either develop something called an alpha helix, which is familiar to anybody who has studied DNA before, right? That's just that kind of corkscrew shape, or something known as a beta pleated sheet, which is basically the chain of amino acids is actually starting to fold in on itself. So if this is the beginning of the chain, this is the end of the chain, there are regions in that chain that are complementary enough that they'll actually create these folds that are held together by the hydrogen bonds. Then tertiary structure is where you're going to end up getting your final three-dimensional shape, and that's driven by the interactions between the R groups. So again, hydrophilic R groups interacting with other hydrophilic R groups, hydrophobic R groups interacting with other hydrophobic R groups, and then also those cysteines interacting with each other to form those disulfide bridges. And now, if you had a protein that consisted of multiple amino acid sequences, you could ultimately end up with what's called a quaternary protein, or a protein that exhibits quaternary structure. So here's 1, there's 2, there's 3, and that looks like 4, right? 4 different polypeptides that each have their own tertiary structure and then join with one another to form that quaternary protein. All right. So these visuals, or these videos will kind of take us through and recap what I just explained.
Most proteins are folded into a complex globular shape. Each protein molecule consists of one or more chains of amino acid monomers. The amino acids are linked by peptide bonds, so a protein polymer is often called a polypeptide. Because they are so complicated, proteins are usually described in terms of 4 levels of structure. Each protein has a unique primary structure, a particular number and sequence of amino acids making up the polypeptide chain. 20 different amino acids are used to build proteins. Theoretically, the various amino acids could be linked in almost any sequence, forming an almost infinite variety of different proteins. This illustration shows some of the amino acids making up the primary structure of a protein. The structure of a single generalized amino acid is shown below. The main backbone of every amino acid is the same. This is what forms the backbone of the polypeptide chain. It is the R group, which projects out from the backbone, that makes each of the 20 kinds of amino acids unique. Different amino acids have different properties that affect the folding of a protein. Thus, primary structure ultimately determines the shape of a protein, which determines its function. In most proteins, parts of the polypeptide chain are coiled or folded, forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the pleated sheet, where the chain folds back on itself, or where 2 regions of the chain lie parallel to one another. Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone are highly electronegative, giving them partial negative charges and leaving nearby hydrogen atoms with partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself. Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall three-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic or polar R groups may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups.
You may want to kind of pause here and play that one back. The tertiary structure, there's a lot going on there. And so you may want to just kind of replay that and try to summarize it in your own words or you paraphrase as best you can so that you can understand that tertiary structure because of just how important it is, right, to the function of a protein. And that'll also, just kind of being well versed in that, will also be super helpful when we do our enzyme investigation, because we're going to be looking into the types of things that can cause an enzyme to denature. And so, if you understand what holds them together, trying to figure out what can cause them to kind of come apart should be made much easier.
Some proteins consist of two or more polypeptide chains. The fourth level of protein structure, quaternary structure, results from the combination of two or more polypeptide subunits. Quaternary structure is stabilized by the same sorts of attractions that stabilize tertiary structure. Hemoglobin, the red oxygen-carrying protein of blood, is an example of a protein with quaternary structure. It consists of two kinds of polypeptide chains. Two of each, a total of 4 chains, make up each hemoglobin molecule.
Yeah, I think I misspoke earlier. I said 6. I think I was thinking about ATP synthase, which is a super important protein we'll talk about in Unit 5 that aids in the production of ATP and is actually a quaternary protein with 6 polypeptides that interact. All right, and then, so that's proteins. Again, the most diverse group of biomolecules that we'll be covering in this unit. And now into nucleic acids, also super important. But the thing about nucleic acids is we'll actually be spending close to a month investigating nucleic acids later in the year. DNA and RNA are the kind of chief topics that we discuss in Unit 7. And so we're really just introducing them here. This won't be a focal point of this unit. It's really just to get your feet wet so that you do understand that this is one of the 4 major groups of molecules required for life. So nucleic acids, DNA and RNA, are information-rich polymers of nucleotides. The monomers that make up these nucleic acids are nucleotides. So like with proteins, the protein itself, the polypeptide, is the polymer. The amino acids are the monomers. Again, carbohydrates and monosaccharides are the monomers. For nucleic acids, nucleotides are their monomers, right? And just to give you a little bit of kind of perspective, nucleotides are actually composed of a sugar. So they actually have a monosaccharide within one of their monomers, right? So that shows you a little bit of relation between the groups of biomolecules. But nucleotides are a bit more complex and bigger than just a simple monosaccharide. So it's a sugar bonded to a phosphate group, which is one of those functional groups that we discussed earlier in the unit, and then a nitrogenous base, one of 4 nitrogenous bases, right? And that's what's going to separate the 4 possible nucleotides from each other. All nucleotides have a sugar. Depending on whether it's DNA or RNA, that sugar will either be deoxyribose or ribose. They will all have a phosphate group, and they will all have a nitrogen base. Just like amino acids, though, something needs to differentiate them. And so what does differentiate them is the nitrogenous bases. For DNA, there are 4, A, T, C, and G. For RNA, there are also 4, but it's A, U, C, and G. So DNA is a double helix, whereas RNA is a single polynucleotide chain. RNA, it turns out, is really just a transcript or a copy of a small portion of DNA, right? So we refer to it as a gene. And so you're only copying or transcribing one of the DNA strands when you make RNA, which is why RNA is only a single polynucleotide chain. Together, they serve as the blueprints for proteins, and thus they really control the life of a cell. So you can have this kind of almost like philosophical argument, what's more important, the DNA or the protein? Proteins do basically everything in a cell, right? Not quite everything, but they are really the workhorses of the cell. But you can't make proteins without the instructions. So, which one is more important? So DNA is the molecule of inheritance, right? That's kind of, it's that's its claim to fame. And then this is an example of a nucleotide, right? So this sugar seen in this nucleotide turns out to be deoxyribose, and it gets its name from the fact that on this carbon here, so I'm going to number the carbons in this sugar, right? This is referred to as the 1' carbon. This would be the 2', 3'. Here's your 4', and then this is going to be your 5', right? And notice that carbons 1, 2, 3, and 4, you don't actually see a carbon there because of the shorthand. But that's just a way to orient yourself so that we know, which carbon we're talking about here. Coming off of the 2' carbon, you only see 2 hydrogens, right, going up or down. But coming off of the 3' carbon, you see a hydrogen going up, but then a hydroxyl group going down. On this 2' carbon, it lacks an oxygen, right? Hence the name deoxyribose, because ribose actually possesses that oxygen there coming off of the 2' carbon. So that would be a ribose. And then if you were to remove that oxygen, that's now known as a deoxyribose. So in DNA, the sugar that you find is deoxyribose. In RNA, the sugar that you would find is ribose. Okay? And it's also important to understand where the other 2 components of the nucleotide are attached, because this is always the same no matter what. Coming off of the 5' carbon, right? Coming off of the 5' carbon, you have that phosphate group, right? Which one of its oxygens is covalently bonded to that 5' carbon itself. And then coming off of the 1' carbon is where you have that nitrogenous base, right? So that 1' carbon covalently bonded to a nitrogen in this base. And this turns out to be the nitrogenous base adenine. You won't necessarily need to be able to identify the bases just based on their structure like that. But later in the year, we'll talk more about the fact that you can identify this base here as a purine because it consists of 2 rings, whereas a pyrimidine like cytosine or thymine would only consist of a single ring, right? So the way that nucleotides are bonded to each other, right, and this is a very, very simplified form. This here is the 5 carbon sugar, ribose. This yellow box would represent the phosphate group that it's attached to coming off of the 5' carbon. And then this here would represent the nitrogenous base that comes off of the other side. And now I'm just making sure. There we go. All right, and that's poorly drawn, but you kind of get the point. So it's phosphate to sugar, which is then bonded to the phosphate of the next one, which is, as we know, bonded to the sugar on that nucleotide, which is then bonded to the nitrogen base coming off in the center here. So making up the outside, or what's known as the backbone of these molecules, is a repeating sequence of phosphate, sugar, phosphate, sugar, phosphate, sugar. So we call it the sugar phosphate backbone. And then the nucleotides are perpendicular to that backbone, facing towards, if this was DNA, they would be facing towards the center of that double-stranded molecule. If it's RNA, then they're not actually bonded to anything. right? So there's DNA, again, sugar phosphate backbone making up the outsides of the molecule, bases making up, what we would call the rungs or the steps in that spiral staircase. Okay, and then what you do need to understand is this, what we'll later really talk about is the central dogma of molecular biology, and that is the relationship between DNA, RNA, and proteins. So DNA and RNA are the nucleic acids. DNA, double-stranded, contains the blueprints for making proteins, but it's housed typically and protected in the center of a cell, in the nucleus or just in the nucleoplasm of a prokaryote. And so you're trying to preserve that DNA because if it's changed or altered or damaged in any way, you only get that one set. And so the way that you go about that is you make copies of specific regions. So that specific region is known as a gene. And when you copy that gene, you copy it into a more usable form, that form being RNA. This happens during the process of transcription. And then that RNA, if you're talking about a eukaryotic cell, that RNA can then leave the protection of the nucleus, head out to a ribosome, and be translated into that amino acid sequence. So the sequence of these bases here in the DNA and the RNA, that's what's going to ultimately dictate the sequence of amino acids. So for example, let's say this gene starts with the sequence T for thymine, adenine, and then cytosine. When you transcribe those three bases in the DNA into RNA, they'll transcribe into A for adenine, U for uracil, and then G for guanine. When you then translate that RNA into the amino acid sequence, anywhere where there is an adenine, uracil, guanine triplet, that will always code for the same amino acid, and that amino acid is known as methionine. Now, you don't need to know that stuff yet, like the specifics of what codes for what, which codon actually leads to which amino acid being placed. We'll talk more about that in Unit 7. Okay, so just a kind of checkpoint question, what role does complementary base pairing play in the functioning of DNA? And I just want to take a second to kind of lay these out. In DNA, wherever you have an adenine on one strand, you would end up having that adenine hydrogen bonded to a thymine on the other strand, right? And it would be hydrogen bonded 3 times. Anywhere where you would have a guanine on one strand, it would be hydrogen bonded twice to a cytosine, right? This is true of DNA. These are the base pairing rules for DNA. For RNA, anywhere where there's an adenine on the DNA and you're transcribing it, that adenine will again be paired with a mistake there. That adenine will be paired with a uracil, right? And that's the key difference here, is that there is no thymine present in RNA. But anywhere where there's a guanine on DNA, that would be paired with the cytosine. Right? And obviously these base pairing rules work the other way around. If there was a thymine on the DNA, it would be paired with an adenine on the RNA. Okay. All right, and then the chapter ends with that further discussion of lactose tolerance that we talked about in class. Definitely something you should spend some time looking into. Those recent mutations that they were able to actually spot in certain populations and the benefits of them, right? So different mutations in DNA have ultimately led to lactose tolerance starting to develop in several human groups. And you're typically seeing that concentrated in areas where you had ancestral connections to dairy farming or dairy cattle. They could take advantage of the abundance of that cow's milk. So researchers identified 3 new mutations in 43 ethnic groups in East Africa that keep that lactase gene permanently turned on, right? Doesn't just turn off, after early life, where you can supplement, a baby's diet with cow's milk. It kind of persists throughout life, and that can be helpful in times of drought, can be helpful, just dietarily supplementing with milk, which is extremely nutritious. I love this checkpoint question, right? Because it really kind of brings the whole unit together. So explain how lactose tolerance involves 3 of the 4 major classes of biological macromolecules. You really should be able to discuss how it involves all 4 of the major classes of biological macromolecules, but just to kind of give yourself a little bit of slack, right? Think about how you could discuss the relationship between 3 of them. Okay. And then there's just the list of goals that you should now be able to, and this is all in your PowerPoint, things you should now be able to do. Now that we've reached the end of this chapter, these are all concepts you should be familiar with, and a way to kind of assess yourself and how far you've come and how far you still need to go. Right? Are you able to do all of these things? And then just a little like practice table. See if you can fill those in without the assistance of your notes. All right. And that's the rest of chapter 2.